I am very happy to give this presentation, and as Lorenzo already mentioned, I work at the University of Amsterdam and the Amsterdam Institute for Global Health and Development, and currently I'm the chair of TBnet for 1 year now. So what I would like to do in the coming presentation is to have a few words about TBnet, and then explain our clinical network and describe some findings that we got from working with our clinical network. And I was specifically asked to share some experience with the implementation of such a clinical network using routine data. So a few words on TBnet. TBnet is a consortium of clinicians and scientists from a wide variety of disciplines, and the main objective of TBnet is to engage in collaborative research in the field of TB. But apart from research, we are strongly involved with capacity building for early career clinicians and scientists, mainly through our yearly TBnet Academy and our European course on clinical tuberculosis. And there's much more information about these activities on our brand new website on the web address tbnet.eu. So this is a little bit of advertisement. So let me start talking about the clinical network. The clinical network was assembled by TBnet while implementing a work package in an FP7 program called TBnet. And the objective of that work package was to describe the clinical management of TB. And to do so, we created a clinical network, which in the end had 23 different health facilities in 16 countries throughout Europe. Within this network, we enrolled 380 patients with proven MDR-TB from all the health facilities in the period 2010-2011. It must be said that consecutively we also enrolled 380 patients with drug-sensitive tuberculosis, but I will not report on this sub-cohort. So the total cohort was actually 760 patients. So the MDR-TB cohort was 37% female. The median age was around 36 years. And surprisingly, more than half of the patients with MDR-TB that were seen through this network were new patients, indicating active transmission of MDR strains throughout Europe. The prevalence of comorbidities like HIV and diabetes were relatively low, and the patients were actually coming from different countries with low, intermediate and high TB incidence, where low TB incidence is less than 20 TB cases per 100,000 populations, intermediate incidence between 20 and 100, and high incidence above 100 per 100,000 patients. And actually that stratum had the most patients in the cohort. And a lot of the graphs and tables that I will show are actually stratified by TB incidence. That's why I put a little bit of emphasis on this. So before going to the results, I really like to acknowledge the project team that beside me was Gunnar Günther, the project leader, and Christoph Lange as the principal investigator, but also the large number of physicians and scientists in the different health facilities. It's not a project that is done by just 2 or 3 people. And lastly, on the last line, we see the names of Cordula Ehlers, Bram Prins and Nelleke Smitsman, who really were instrumental in support in the administrative area and data management. So let us talk about some results that we got from the clinical cohort. First, I want to discuss drug resistance. And this first graph actually shows the coverage of drug sensitivity tests at the start of therapy. And we can see that from the usual drugs like ethambutol and pyrazinamide and even the fluoroquinolones and second-line injectables, the coverage of drug sensitivity testing at the start of therapies was 90% or above. And we can actually say that in the total cohort, 90% of the patient had at least one drug sensitivity test for one second-line drug. And the differences between the different strata of TB incidence were not very large. It's a slightly different story when we look at drugs used for the more complicated MDR treatment regimens, where we see that although ethionamide and prothionamide still had an average coverage of 90% or above, the coverage was really low for drugs like cycloserine or PAS, and more or less non-existent in high incidence countries for a drug like linezolid. So drug sensitivity testing at the start of therapy is done for usual drugs, but not for the more complex drugs. And when we look at those more routine drugs with a high coverage, we see that if you test for them at the initiation of therapy, you will find relatively high prevalence of resistance, more than 50% for ethambutol, 40 to 70% for pyrazinamide, a little bit less for the fluoroquinolones and the injectables, but almost 40% or more of the patients had at least additional resistance to one second-line drug in the MDR-TB cohort. So that means that when we talk about MDR-TB cohort, we actually talk about a cohort with a lot of additional drug resistance, not only for the fluoroquinolones and the injectables, but especially like drugs of ethambutol and pyrazinamide. And these drugs play an important role now, for example, in the Bangladesh regimen. So our MDR cohort was actually an MDR plus cohort where more than almost one third of the patient had proven pre-XDR or XDR-TB. So that should be kept in mind when we discuss further results. So then what we try to do, that if we know that there is additional resistance in a typical MDR cohort, would we be able to predict this additional resistance within a cohort? So what we see here is that the presence of a comorbidity like HIV, hepatitis, of diabetes actually increased the probability that there was not only MDR but additional resistance, so pre-XDR or XDR. And that was an association not seen for the typical individual risk factors like sex and age or having had previous TB. Although interesting, we must be very careful with interpreting this data, because as you can see, the confidence intervals around these estimates are rather wide. But we thought it was an interesting initial idea that there might be some association with concurrent comorbidities and additional drug resistance in an MDR cohort. So while we were collecting all our data on resistance and outcomes, there was this guidance by WHO on the use of short course MDR regimens. According to several criteria, people were either eligible or non-eligible for such a short course regimen, and the main criterion was actually proven or suspected susceptibility for all the drugs within the regimen. So we took the opportunity to assess if the TBnet cohort could provide data on how many people were eligible for a short course regimen. And what is this short course regimen? It is actually constructed around ethambutol, pyrazinamide, moxifloxacin and clovesamin, but it has additional drugs in the intensive phase being high-dose isoniazid, kanamycin and proteonomide. Now, given the previous results that we showed on the coverage of drug sensitivity testing and actually drug resistance, it might not be surprising that in our TBnet cohort there was a very low percentage of patients that would be eligible for such a short course regimen. Because first of all, from all our patients included, only 148, so 39%, had actually drug sensitivity tests done to assess the criterion of susceptibility. And of those people, only 18 had proven drug sensitive for all the drugs included. And we took the opportunity to work together with different cohorts at the same time, and this low percentage is really corroborated in every single cohort that was providing that data at the moment. So eligibility for short course MDR regimens in Europe is very, very low. So what about drug resistance during therapy? But the first observation was that although everybody was an proven MDR patient, around 20% of the patients never had any DST done after starting treatment. So that was a finding in itself. When it was done, we could see that there was overall 17% of the patients showed additional drug resistance, and that could be even to 4 or 5 new drugs that showed resistance. But there's also a marked difference in the strata of TB incidence. So acquired drug resistance being relatively low in low incidence countries, but reaching up to over 20% in high incidence countries. So the topic of resistance we can conclude as that there is active transmission of MDR strains, that apart from drug sensitivity testing for fluoroquinolones and second-line injectables, testing for key second-line drugs is not very frequent, and that especially there are very large difference in the frequency of drug sensitivity testing during treatment. But if you treat, you find a marked frequency of acquired drug resistance, and that holds especially for countries with intermediate or high TB incidence.